Okay, so I think now we can start. This meeting is being recorded for those of you that didn't figure that out in the beginning. And I was just thanking Sylvia for inviting me and Joao for making my stay here so pleasant. So before I start talking, I usually don't like spending all the time talking by myself during a presentation. So please take out your phones and either scan the QR code or enter one of these two websites, because all throughout the presentation, actually, there will be just 4 questions in the presentation. The first one, which is the easiest difficulty, during which I will be assessing kind of your level of understanding of what's going on, and also which will allow me to have a breather. So whenever you will see a black slide with the direct poll logo, you will know that it is time to take out your phone. Don't close the browser with the link that you have just opened, and you will be prompted with a new question. That way I can kind of judge what is the temperature of the room. You will always have 1 minute to answer the question. That way I will not be waiting forever for everyone to fill in the responses. So I guess you have already accessed the very first question. It might be a little bit less than a minute that I have given you, but let's just check the results. Also, the people which are at home, you can also scan the code and participate in this. You don't have to be in the room in order to do it. So I just try to sneak in a few sci-fi and fantasy references throughout my presentation, and this slide already has one of them for those of you that are fans of Sir Terry Pratchett. So now let's go to the actual scientific part of my talk, which is called Beyond Selfing versus Outcrossing, or the environment-dependent benefits of mixed mating. Now, I wanted to introduce myself a little bit because this is partly for hopefully will establish future collaborations with colleagues here. So I come from the University of Ostrava, and I'm in general interested in the evolution of plant mating strategies in response to environmental variation. So these are the plant species with which I have worked so far. Some of them are annuals, some of them are perennials, but most of them make pretty flowers. Among the 2 big projects that I kind of have ongoing, about which I'm not going to talk into detail today, is a life form Inuartia project that is in collaborations with colleagues from the Charles University in Prague, and where we study the effect of habitat degradation on genetic diversity and inbreeding depression in an endangered serpentinophyte species. So the species only exists in 2 locations in the Czech Republic and nowhere else in the world. And I was studying how its genetic distribution is partitioned among habitats, how related it is to other species from the same complex, and also evaluated how inbreeding and outbreeding depression vary between the habitats, with possible, which will provide insights if we decide to reintroduce the species in places where it went extinct. Another project that is just in the beginning, maybe you recognize all of the pop culture references, the TARDIS, the DeLorean, and the Time Turner, is called Time Capsule, because it kind of does ecological time traveling for the study of rapid evolution and ecological epigenetics using a novel approach called resurrection ecology. Currently, I'm only focused on a species that is called Centorium erythrae, but I hope this to become a multi-species, multi-climate project. And the idea is quite simple behind it. I have seeds that have been stored in seed banks for at least 2 decades. And I went and resampled the seeds from the exact same populations in 2020, and I will be growing the ancestral and descendant populations in a common garden. And from there on will stem a lot of other consecutive analysis. The idea is to see if and how plants adapted to climate change and pollinator decline over the last 2 to 3 decades. I'm also quite involved into popular science and science communication. All the way in the back here is me with the team of Science for Children in Macedonia at the very first scientific conference for children in Macedonia that happened this year. This is the materials that we used for a workshop on heterostyly. So some people might be interested in this here. And I'm missing only 800 subscribers to get a YouTube button, because I also have a YouTube channel where most of my teachings are freely available under a Creative Commons license. Okay, so Let's go straight into the topic of the presentation today. And the people which are here are actually not able to see the titles. That's going to be fun.
At home, we don't see it.
Sorry?
At home, they don't see it.
At home, they don't see it, but here I will have to read the more.
There is high
Float, yes, excellent. So now you can see things nicely. Let's talk a little bit about mixed mating strategies, or rather mixed strategies in general, which occur in species that use two or more different tactics with different consequences for their fitness. So just a few examples that are not all plant related to show you that I also can look into things that are not plants. Here are the leaves of eucalyptus that is very abundant in here, which surprised me, that change their shape as the plant gets older in order to favor the exposition to sunlight. You have the this defensive, the one with the spike, and non-defensive phenotypes in the water flea, Daphnia magna, that is induced by predation. So obviously the spike grows when there is predation in the environment, and for energetic reasons, it is not developed when there are no predators in the environment. We have also this left and right feeding strategies of scale-eating cichlid fish. I'll talk about them in a detail just in a minute. We have obviously the heterostyle, and the one thing on which I will be focusing mostly today will be dimorphic cleistogamy. Now, what is very interesting about the mixed mating strategies is that they can be maintained in an environment thanks to a selective mechanism which is negatively frequency dependent. What this means is that the fitness of either a tactic, there can be two or more, decreases as its frequency in the environment increases. So a historic and very well-known example is the change in the frequency and the fitness of the two feeding strategies of the cichlid fish. So we have the right-bent fish, which are particularly well adapted to attack their predator, of which they nibble the scales from the left side. And then the left-bent fish, which are very successful at attacking their prey from the right side. They tend to mix left and right, so hopefully I will not confuse you altogether with this. So what happens is that if the frequency of either of these strategies is too abundant in the environment, the prey is going to start to be vigilant on one side of their body in particular, which will then in turn decrease the success of the attacks of the scale-eating cichlid fish. This means that the frequency of, let's say here, the left bent strategy is going to change over time as its fitness changes, and the right bent strategy is going to be complementary to it. And these two frequencies tend to vary in time until they end up reaching some kind of stable equilibrium. And in this case, it should be about 50-50% of each strategy. Now, mixed mating strategies combine two modes of reproduction. One is selfing or self-pollination, and the other one is outcrossing or cross-pollination, where the pollen is brought from another individual of the same species. In plants, this happens thanks to pollinating vectors, if the plants are entomophilous, obviously. So a mixed mating strategy can happen at the level of a single flower, at the level of a plant where different flowers have different mating strategies, or at the level of the population where different individuals have different strategies. Now, what is very important is that each of these strategies has certain advantages and disadvantages, or as I would rather call it, characteristics. The most important advantage, the biggest benefit for the fitness of the selfing strategy is that when an individual reproduces with itself, it leaves two copies of its genes to its offspring, one through the pollen and the other one through the ovules. And when an individual outcrosses, it only can leave one copy either through the pollen or through the ovule that has been pollinated from external source. This is what we call automatic transmission advantage of selfing. Selfed individuals, if everything else is the equal between the two strategies, simply transmit twice as many genes to their offspring than outcrossing individuals, strictly outcrossing individuals. And this automatic transmission advantage can be diminished or even canceled out if there is inbreeding depression. Inbreeding depression is simply defined as decrease in the performance of inbred, in this case, selfed individuals, compared to outbred individuals. So we can look at this as qualitative versus quantitative advantage of the two strategies. In outcrossed individuals, there is no inbreeding depression. In self individuals, there could be high inbreeding depression. Now, if only these two forces are acting in a population where the two types of mating are encountered, mixed mating cannot be a stable strategy. And this is because there is a positive fitness dependence created by the automatic advantage in absence of inbreeding depression. This is a very fancy phrase, so the simple way to see at it is that as the frequency of selfing individuals increases in the population, their fitness is also going to increase. And as a consequence, the fitness of outcrossing individuals is going to decrease because the outcrossing individuals have fewer and fewer partners to reproduce with. And this means over time, if there is only the automatic advantage and no inbreeding depression, the selfing strategy is going to invade the population and the outcrossing strategy is going to disappear. On the other hand, if we have now automatic advantage, but also high inbreeding depression, let's say that this is the fitness of the outcrossed individuals, but the self individuals suffer such hard inbreeding depression that they all completely die out before even being able to germinate. In this case, we will have the outcrossed offspring, the outcrossing strategy that is going to invade the population, and the selfing strategy, which is going to disappear from the population. Now, what is interesting is that in 2005, there was a study compiling almost 350 species and their estimates of selfing rates, and they showed that about 50% of the individuals are either predominantly or completely selfing or predominantly or completely outcrossing, but about half of the species for which we had estimates of the outcrossing rate had a mixed mating strategy. This is way too big of a number in order to support the hypothesis that either complete selfing or complete outcrossing are evolutionary stable strategies. So we need to find other ways to explain the maintenance of mixed mating in a population. And one of these explanations is the existence of environmental variation which can maintain mixed mating. So if an individual or a population experiences different environments, sometimes which favor self-pollination and sometimes which favor out-cross-pollination, mixed mating can be a possible stable strategy. Among the environments, variable environments that can favor these two strategies is obviously the abundance of pollinators, can be the abundance of resources, and can also be the intensity of inbreeding depression. We often tend to think of inbreeding depression as a characteristic that doesn't change and it only depends on the coefficient of inbreeding of the individual. But in reality, inbreeding depression is also dependent on the environment in which the individual is raised. So how does this plastic mixed mating work? Well, you have let's say two options. First, you can have a fixed mixed mating strategy, which means the individual always has a certain percentage of selfing and a certain percentage of outcrossing that do not change regardless of the environment. This is quite advantageous when environmental variation is random and unpredictable. So this fixed mating strategy is also called Bet-hedging. And if you have ever gambled, this is kind of a way to increase your chances to win. Now, if you do this kind of Bet-hedging strategy to win to roulette, this is not going to work out. You're definitely going to lose money. But at least with this kind of strategy, you're sure to never lose because you're always having either a little bit of self or a little bit of outcrossed offspring that can survive in whatever environment they encounter. But if the environment is predictable, such as, for instance, the seasonal variation of pollinators that are more abundant in long and warm days compared to short and cold days, then a very good strategy is to be plastic and mixed mating, which means that the proportion of self and cross-pollination can be adjusted in response to reliable environmental cues. Now, one example would be plants that are capable of decreasing the distance between their anthers and stigma if the flowers are not successfully pollinated. And another example is the plastic cleistogamy in Lamium amplexicaule and other cleistogamous species. So let's get into cleistogamy finally. Cleistogamy is the ability that some plants have to produce these closed flowers that cannot receive pollen, so they have to self-pollinate, they're obligately self-pollinating. But most cleistogamous species also produce chiasmogamous or open flowers that are potentially outcrossed. These flowers are usually hermaphroditic and don't have a self-incompatibility system, so both self and cross-pollination are possible within the open flowers. Cleistogamy is a trait that has evolved over 20 times among different angiosperms, and in different families can confer different fitness advantages, but I will be mostly focused on one species today. Just a little overview of the different types of cleistogamous species that exist. Here you have a species where the open flowers are produced on the aerial part, so very accessible to pollinators, and the closed flowers are produced on underground stems, so definitely no access to pollinators. In other species, such as the star of today's talk, Lamium amplexicaule, or Scutellaria leonardi, the open and closed flowers are produced at the same level and sometimes at the same time. In Viola pubescens and other violets, in cleistogamous species, the open flowers are produced on the top of the flowers, like high on the flowering stems, whereas the closed flowers are produced on horizontal stems close to the ground, because again, they don't care if they attract pollinators or not. And you also have aquatic plants where obviously open flowers are above the water, closed flowers are below the water level. So what is interesting about cleistogamy is that it has the potential to be a mixed mating system. In Lamium amplexicaule, for instance, the proportion of open flowers, which is this percentage CH, it's a notation that I will use consistently throughout the talk, varies between 25–30% to 0 up to 15%, depending on the environment. So when the days are warm and long, such as spring, and the pollinators are abundant, the plant tends to produce more open flowers. And when the days are cold and short, the plant produces more closed flowers. What is also very interesting is that the proportion of open flowers is determined in advance. It's not a last minute decision of, oh, the day is nice, so I'm going to open my flower. The plant has to be exposed to the environmental cues up to 2 weeks prior to flowering in order to determine the production of open and closed flowers. And another very interesting thing about cleistogamy, and then I will go into my own results, is that sometimes the reproducing through open and closed flowers goes beyond the genetic effect of inbreeding and outbreeding, because there can be some non-genetic effects on the fitness of the seeds. In some species, especially the ones that have these very inconspicuous closed flowers, there is a huge difference in seed size, which means that the seedlings already have some kind of advantage from the very start of their germination. But in other species, there are no visible differences in the seeds, even when the seeds are produced from self-pollination, whether it is open or closed flowers. And yet we see significant differences in the performance of the plants from open and closed flowers, even after we control for the degree of inbreeding. And there can be some non-genetic epigenetic effects on the fitness. So let's talk about Lamium amplexicaule, which is a cleistogamous species, in case you haven't guessed already, that has this specific mode of flowering where it always starts by producing only closed flowers. And this is what is called constitutive cleistogamy. And then depending on the environmental conditions later in the season, it produces a mixture of open and closed flowers in variable proportions. This is what I call induced cleistogamy. So I will present you the results of 3 independent studies that I have conducted over the course of 10 years with objectives that are complementary, but I was slowly improving my research in order to be able to answer more and more complex questions. So I'll start with something very simple, which is to study the relation between cleistogamy and mating strategy. Is variation in the proportion of open flowers actually a variation in the mating strategy of the plant? Then I have studied the variation of cleistogamy in different environments and see whether it is plastic or a Bet-hedging strategy. And lately, I evaluated the adaptive character of plastic cleistogamy in response to different hypotheses of what could maintain it in the nature. So let's go with the first study of the relation between cleistogamy and mating strategy. It's important to know what is the outcrossing rate of the open flowers, and also does it vary between populations and different environments. And then I would use this information to find out whether the outcrossing rate of the plant as a whole varies between environments. So the overall question is dimorphic cleistogamy equivalent to mixed mating? If it is, this is very advantageous because it means that we can approximate the outcrossing rate of a plant or a population by simply knowing the proportion of open flowers. So to estimate the outcrossing rate, when you don't have these informations, it's a little bit trickier because first you need to be able to estimate the outcrossing rate of open flowers. And for this, it's a pretty straightforward task. You can calculate the number of individuals produced by cross-pollination and divide it by the total number of individuals produced by self and cross-pollination. Now, this is a straightforward task only if you know the type of pollination from which the seeds were issued, which is not always easy to determine, but let's put it that way for now. Now, this is all you need to know when you determine the outcrossing rate of a cleistogamous species with only open flowers. But you shouldn't forget about the proportion of closed flowers in the population in cleistogamous species. This means that in cleistogamous species, the total outcrossing rate is proportional to the outcrossing of open flowers and the proportion of open flowers. Now, I did an introductory course about calculating outcrossing rates with progeny arrays earlier this week. And it turns out that this might use a little bit more explanation. So it is very simple. If you know that a plant produces 50% of open flowers, the flowers are 100% outcrossing when they're open, so there is no self-pollination, then the total outcrossing rate of the plant will be 50%. If you need extra explanations about this, please let me know. We did waste a lot of time explaining this on Tuesday. Okay, so what I did, this was work during my PhD, is I sampled 4 populations in France. 2 of them were in Dijon, in northern France, where the climate is rainy and continental, and 2 of them were in Montpellier, in the south of France, where the climate is hot and dry. In each of these 2 regions with contrasting climates, I was able to sample 2 populations about 5 to 6 kilometers apart. And one of the populations always correspond to a population found in a large habitat, which is the typical characteristic habitat of Lamium amplexicaule, fields or vineyards that have been tilled early in spring. Lamium amplexicaule is not a competitive species and it thrives in this type of habitats. The populations are large, bearing hundreds and even thousands of individuals. And each plant is quite impressive. It has dozens of flowering axes. And then on rare occasions, Lamium can also establish in these habitats that are densely colonized by other vegetation. But in this case, the populations are not very happy. These kind of small populations usually bear very few individuals, 10, 30, maybe 50 at maximum, and the individuals usually have only 1 flowering axis. So let's get your phones ready. I think you had a glimpse of the dark slide. But before I show you the results of my estimates of outcrossing rates and production of open and closed flowers, can you tell me what results do you expect based on what I have told you about Lamium so far? Should the open flowers in unfavorable habitats outcross more? The open flowers in favorable habitats outcross more, or no effect of the environment? And then same thing about the production of the open flowers. So ideally, you would give me 2 responses. Choose one of the 3 options on the top and one of the 3 options on the bottom. And I will have 1 minute of rest. One minute is a very long time when I have to stand in front of you all. It's not working? Then it will be even more than 1 minute. There you go. Let's start again. 1 minute 30 of rest for me. Lucky me. I'm not showing the poll results as not to influence you and so that you would copy from one another. In 5, 4, 3, 2, 1. I don't know what the people at home are doing. Hopefully they're not bored by now. Okay, so there were no right or wrong answers since you don't have the input data that I have. But the winners seem to be that the outcrossing rate should be higher in favorable habitats, because we said that when the resources stimulate plant growth, they should also favor cross-pollination. And it's a tie between percentage of open flowers being higher in favorable or in unfavorable habitats. Quite interesting. You notice that I didn't talk about the climate at all, so I guess that can give you a hint of what happened. Now let's switch the slide. Okay, so let's start with the outcrossing rate. On the left, you have the populations from the south, and on the right, you have the populations from the north, colder and rainier weather. In yellow are populations from favorable habitats, in green populations from unfavorable habitats. Now, there is some variance in these estimates, and you see here that the value for the northern small population is quite low, but this is probably due to the fact that the population was highly monomorphic. So with the method of estimating outcrossing rates that I used, it was simply impossible to distinguish a selfing from an outcrossing event because the fathers had all the same genotypes as the pollen receivers. So you'll have to take my word, I will not show you all the data and calculations I made, that we will assume how the outcrossing rate is actually constant between favorable and unfavorable environments between the south and the north of France, which is quite neat because from this point on, I can always go into a population and assume that about 25% of the seeds produced by open flowers come from outcrossing. Now, let's see what happens with the production of open flowers. Again, I tend to mix up left and right, but this is not it. The yellow populations from favorable habitats produce fewer open flowers proportionally compared to the populations in unfavorable habitats. And this can be a surprising result, but it is consistent again between habitats from two very contrasting climates and populations that are 400 kilometers apart. So what does this tell us about the initial questions that I asked? Is dimorphic cleistogamy equivalent to mixed mating? Well, we can definitely deduce the outcrossing rate by simply estimating the proportion of open flowers in the population. And this is possible because the outcrossing rate of open flowers does not vary between populations, habitats, and contrasting climates. So what is the outcrossing rate of the plant and does it vary? Well, the total outcrossing rate seems to increase in unfavorable habitats. This could mean that having a little bit more of outcrossing in unfavorable habitats could be done to increase the adaptive potential in such habitats by increasing the overall genetic variation in the offspring generation. But more about it at the end of the talk. So the next objective was to study the variation of cleistogamy in different environments. So we now know that in favorable habitats, the populations have higher proportions of closed flowers. In unfavorable habitats, the populations have higher proportions of open flowers. But this is what I observe in natural populations. And I don't know if this variation is, for instance, due to phenotypic plasticity or genetic differentiation between the habitats. And also, I don't know which environmental factors specifically favor the production of each floral type. So I jumped at the occasion during my PhD to immediately harvest the seeds from natural populations and conduct a common garden experiment in which I stimulated the favorable habitats by removing all the vegetation in a soil strip with tilling, and the unfavorable habitat where I just left the vegetation intact as is. So here is the color code that I'm going to use. Southern populations are with full lines, northern with dashed lines, and favorable habitats in a non-competitive environment are in yellow here. Just as a reminder, this is what I observe in the natural populations. And now let's see, surprise, surprise, what I observe in my experimental environment. Let's start with the positive things. First of all, there is some kind of genetic differentiation because the small southern population here seems to produce a markedly different proportion of open flowers across both environments compared to all others. This could be due to genetic differentiation. And there is also phenotypic plasticity because the production of open and closed flowers varies between the two treatments. However, it varies exactly at the opposite of what I have planned. Now, I have made this presentation having students in mind since I'm here on a teaching exchange, and I just wanted to dedicate a couple more minutes on explaining what happened afterwards. What happened is that I was a very impatient PhD student and I was in a hurry to have this experiment finished as soon as possible. So I planned everything out nicely, but I also planted the seeds immediately after harvesting them, which was kind of in late April, early May in the south of France. So this is the temperature in the natural population sites during their actual flowering seasons in the south. This is the temperature in the north, and this is the temperature that they had on my experimental south in mid-June, 5 to 10 degrees higher. And if you look at the precipitation levels, it's even more disastrous. So I absolutely didn't account for an extra level of climatic variation. And at this stage in my PhD, I could only conclude that extreme drought and heat severely impede the production of open flowers because they simply stunt the growth of plants. That's it. So, and then I kind of ran out of funding for this project and had to move on to the next questions of my PhD. And I left this on a pause for about 7, 8, 9 years. That is until 2019, when I was like, okay, so I know that there is something about the competition. There is something about the climates. Let's see how we can put all of these things together. I couldn't access the French populations, but I was able to find 7 natural populations that were distributed along a climatic gradient in 3 countries, continental in the Czech Republic, Mediterranean in Macedonia, North Macedonia now, and desert in Israel. And whenever possible, in each of these climates, I tried sampling populations from open and closed habitats. And when I say I, it was mostly nice collaborators that did this. What I did is then I took the seeds and I did another common garden experiment in Ostrava, the Czech Republic, so highly temperate, very rainy climate, which means that I was definitely not going to have problems with missing rain, but I was going to have problems with too much rain. So I had to construct these PVC roofs that would let all the light come in, but prevent the rain from falling. And I ended up with a full factorial design of competition and drought. So I have treatments with competition, without water, and with water. I expect this is how these treatments are going to influence the growth of my plants. So the most favorable environment with no competition and as much water as needed will enhance the plant growth. The most competitive environment in which plants are severely limited in water will stunt their growth, and the 2 other treatments will be somewhere in between. And again, a question for you. Let me start the poll first. There you go. What do you expect will happen in this experiment? Is competition going to increase the proportion of open flowers, decrease the proportion of open flowers, same thing for the drought? Or do you expect some kind of interaction between competition and drought that are again going to make the results very funny to interpret? I'm not sure time-wise, since we started later, can we shift it a little bit more? Okay. I mean, this is the most interesting part of the talk anyways. After I'm done with this, it's like, I have some other stuff, but they're not as silly as this one. Oh, yes, I didn't do this by chance. But also there is a great YouTuber that has a channel called Veritasium saying how people tend to retain more the explanations if they have tried to answer the question by themselves first. So I don't have a control group, but I'm giving the same talk next week. Maybe I can compare the results afterwards. Okay, time is up. Let's see what's going on. So let's start with the plant size. I hypothesize that competition and drought will stunt the growth of the plants. No, I'm getting there. Okay, so the results will show in a second. But just to show you that on the left will be the treatment favorable one without competition, on the right with competition. And in blue here are the plants that have received sufficient levels of water. So clearly, when plants receive water and grow without competition, they're doing very well. And when they receive water but have high competition levels, they're not doing so fine. And this is what happens when the plants are exposed to drought stress. It does reduce to some extent the growth of the plants in the non-competitive environments, but we see that there is also an interaction between competition and drought. And this is simply because once the plant has one flowering axis, it can't really grow smaller than that. So that's it. And now let's see what happens with the proportion of open flowers. In the treatment where the plants receive sufficient amounts of water, finally, I have a small yet significant increase in the production of open flowers when the plants are facing competition, same thing as I observed in natural populations. And this is what happened when I add droughts. It's actually fairly consistent with my initial hypothesis that if you dry the plants out to the extent that they're really struggling for survival, they will only produce closed flowers. And another thing important, I will insist on this once more, is how the size of the plants does not determine the production of open or closed flowers. I will get back to this in a minute. Before going to the conclusions of this, I just wanted to show you the part related to the population genetic differentiation. This is the answer, the phenotypic response of the plants when they receive sufficient amounts of water by population. And here is this one. This is the Israeli population that grows exactly on the edge of the desert that seems to be producing open flowers no matter what. It always just produces open flowers and is very happy doing so. So there is clearly, or hopefully, some kind of ecotype that doesn't exist in Europe, but exists in Israel. Unfortunately, I no longer have seeds from this population. So, back on the original question, what determines the variation in cleistogamy? Is it phenotypic plasticity or genetic differentiation? Well, it is both. And we do have a possible existence of ecotypes. And then which factors favor the production of each floral type? Competition, drought, and their interaction together all influence the production of open and closed flowers, but definitely not plant size. And what is probably happening, although I haven't tested this on the physiological level or the plant regulation level, is that you remember the plant always starts by having this constitutive cleistogamous proportion, which means that if the plant then reaches favorable enough conditions for its growth, it will switch to the induced cleistogamy, where the production of open and closed flowers will depend on the environmental conditions, which is quite neat. Finally, with 10 years of experience, I managed to get some results that actually correspond to my initial hypothesis. This is to encourage the students never to give up. So now that we know all this, remains still one question. We know how patterns vary. We know that cleistogamy is kind of an adaptation of mixed mating, but is this actually adaptive? And this is what I'm going to try and answer in this last part. So I have emitted two hypotheses, but we actually already have the data for, I've already presented you the data for testing 3 hypotheses, which we will see in the end. So is cleistogamy adaptive to environment-dependent inbreeding depression or to variation in the pollination environment? We will probably skip that part. And to do this, this was an experiment that was actually running in parallel with the observations in natural populations, so I don't have exactly the same data. But in one population, I made hand pollination of Lamium amplexicaule, and Katerina and Sarah know how much I hated that, where it was easy for the self-pollination of closed flowers. I just let them do their thing. They were very happy to produce seeds. Then I carefully took the pollen from the plant and deposited it on its stigma in order to produce self-pollination of open flowers. And I also cross-pollinated open flowers by hand, but I had to castrate them first. And also to test for the effects of castration, I also had to castrate the selfing flowers. And trust me, if you're a PhD student, it's a great experience. You should do it, but not otherwise. And now, because the population that I studied here actually is able to produce up to 2 generations a year, one in the spring and one in the autumn with variation in the production of open flowers, I grew half of the seeds in autumn and then the other half of the seed lot in spring and saw what is happening between them. So you know what comes next? Oh no, what comes next is, because maybe you will ask me the questions, I didn't observe any statistical differences in the size of the seeds from the different cross treatments, in the germination rates of the seeds, or in the survival rates of the seedlings, in case you were wondering about this. And now is a question for you. What do you think I have observed as results? Did I detect inbreeding depression, meaning that when you are a self individual, regardless of the flower type, your fitness is decreased? Outbreeding depression, where outcrossed individuals are less performant. An effect of the flower, where being issued from open or closed flowers affects your fitness, regardless of your degree of inbreeding. An environment-dependent effect, or no effect of the environment, experimental treatments at all. And I forgot the timer, but it's better for me. Your results. Right. Well, I did have my break anyways. So yeah. But as you clearly saw that this is just an excuse for me to have a break. So I actually can go back and we will see what it says. Four, 3, 2, 1. Let's go to your results. Okay. Yeah, of course, you all voted for the environment-dependent effect. Some people were talking about inbreeding depression, some were talking about outbreeding depression, and nobody was talking about flower effect. That is very interesting. You already know what's going to happen. Again, the different colors here will correspond to the different cross types. These are open flowers, the yellow one is closed flowers. And on left, you have the spring season. On right, you have the autumn season. What happens with the production of open and closed flowers is that in spring, offspring of open flowers, regardless of whether they're inbred or outbred, produce more open flowers. These actually denotate statistically significantly different results. And in autumn, we see a clear decrease in the production of open flowers, and also there is no difference between the 3 types of offspring. It doesn't matter whether you're from open or closed flowers, they all have the same mating strategy. Now we are interested in inbreeding depression, so what happens with the fitness? Well, again, we see that in spring, individuals that are issued from open flowers have higher fitness than individuals that are issued from closed flowers. So spring favors the production of open flowers overall, and also favors individuals that have been produced by open flowers. One important thing at this point, I said very interesting, nobody selected flower-dependent effect. You see that here we have 0 difference between inbred and outbred offspring from the open flowers. So there is no inbreeding depression, at least that we can detect with this experimental assay. But we do have an effect of the flower. And now if we look at what happens in autumn, everything, like the patterns are completely inversed. We no longer have grouping of the results based by the flower type or by the degree of inbreeding. So it's quite interesting, but I'm not going to go into details with this because we don't have time about it. And to be honest, I'm not even sure what's going on myself. So is plastic cleistogamy an adaptation to environment-dependent inbreeding depression? The short answer is no, because we simply don't detect any inbreeding depression. But there is a consistent effect of floral type, at least in the spring environment, where plants being issued from open flowers tend to have higher fitness than plants being issued from closed flowers. And what could be happening here, in the beginning, I was talking about this possibility that when individuals have the same degree of inbreeding, but are produced by open or closed flowers, they have different fitness. And this is definitely not due to genetic effects, to heritable genetic effects, but could be due to heritable epigenetic effects. And ideally, sometime in the future, it will not be in 2023, because earlier this week I found out that I'm not getting money for this. I will assess the variation in methylome and its effects on cleistogamy and fitness in Lamium amplexicaule, and I will keep you posted if you're interested in finding this out. So we're going to skip the adaptation to variation in the pollinator environment because it's already quite late and it's Friday afternoon after all. But the animations are a bit long. And let's just go to the conclusions and perspectives of my work. So I had 3 objectives: assessing the relation between cleistogamy and mating strategy, determining what causes the variation of cleistogamy across environments, and assessing the adaptive character of plastic cleistogamy. Regarding the first objective, the most important conclusion is that, at least in the French populations that I have studied in spring, the variation in the proportion of open flowers is equivalent to variation in the total outcrossing rate. So usually when I now go and study Lamium and amplexicaule in natural populations, I just count the amount of open flowers, count their ratio, and then I know how much the population outcrosses. Now, the variation in different environments seem to be plastic and influenced by drought, competition, season, and their interactions. And there is also some genetic differentiation, which I'm planning to study again whenever I get the funding for it. I don't have the genetic differentiation part, but for the last part, so is plastic cleistogamy an adaptation to variation in resource abundance? Probably not, because we saw that plants with different sizes do not necessarily produce the same patterns of cleistogamy. Is it an adaptation to inbreeding depression? We don't detect any inbreeding depression, so no, at least in the crosses that I tested. Is there an effect of the floral type? Could this be something that provides an adaptive advantage? Could be. Again, need to test it more. And you have to take my word when I say that it could be also an adaptation to the pollination environment, simply because open flowers tend to outcross more in spring when they're more frequently produced. Among the future directions that I would like to test is to see how competition-dependent inbreeding depression and flower affect the fitness of the plants. This is a handful. There will be controlled pollinations in different environments, but also they will be done in different populations, hopefully including different ecotypes, such as the Israeli one or the small population from the south in France. And I would also like to test the effect of epigenetic variation on the expression of cleistogamy and its fitness using pharmacological demethylation assays, for which I will not need any extra funding and I will start them in spring, and also using bisulfite sequencing, which obviously will be a little bit more costly. And with this, I would like to thank you for your participation and for your attention. In case you're interested in knowing more about what I have done, not only on cleistogamy, but in my other studies, the QR code is actually my virtual visit card. Yeah, visit card. I also have real visit cards if you would prefer them that way. And just a small shout out to my friend Natalia Aguilera Lopez that did the amazing illustrations I have been using throughout. Thank you. Do I need to stop the recording now?
We can do like questions now.
Yeah.
We can record it as well.
Okay, I just don't know how to stop. Yeah, there you go, screen sharing. Okay. It looks like I haven't shared the screen all throughout.
We were at home, they were seeing very well.
Okay, well, in that case,
Everything went well.
great.
So we can go for questions. We can start in the room, but then anyone at home can also pose any question. Does someone like to make something?
I have a question. I don't know if I need to go there completely at home.
I can repeat it, yes.
So congratulations, it was a huge amount of work. It's very interesting work. And I was actually having a hard time to follow because it was a lot of information very important. So maybe that's why I didn't get that last part, but you showed the one of the last slides that the self-pollination causes different effects depending if they were open flowers or closed flowers.
Absolutely.
And I'm not getting that. So how do you expect that maybe on the same individual who produces open flowers and closed flowers, the fact that the flower is open or closed, if you guarantee that it is self-pollinated flowers, the seeds will have an offspring that behaves differently?
Exactly.
That's a great question.
Exactly, absolutely. I mean, this was a study that I did, like I had the results in 2010 in my second year of my PhD, and I only published them in 2019 because back then, like heritable epigenetic variation was not a thing, and it was just impossible to convince reviewers that this can be something. I don't know about the specific mechanisms. I don't know what causes this. All I know is that I have seen this pattern. My first step would be to try and reproduce this using other populations to see if we get the same results in other populations with open and closed flowers. And then given how the production of open and closed flowers is, for instance, regulated by a hormonal ascending flux, I think it's, do we need the slide? Okay, it's a gibberellic acid, GABA? No, gibberellic acid that it regulates the opening and closing of the flowers way in advance. So I would imagine that the concentration of the hormones is also different in the flowers at the time of pollination and seed maturation. Maybe that has an effect. Maybe there is some kind of epigenome modification that happens only in open or only in closed flowers and is transmitted to the offspring. I have no clue. So in addition to testing this with other populations, the very simple experiment I'm going to do is take self-offspring from open and closed flowers in which I know there is difference in fitness, and then subject them to a demethylation treatment that is supposed to diminish the differences in methylation levels. And if there is some kind of epigenetic effects, I expect these differences to be cancelled out, the difference in fitness to be cancelled out after demethylation. But this is something I have had in the making for a while, first pending a funding, then pending for COVID to be over because I lost one generation of hand pollinations due to COVID. So anytime now, well, anytime like in the next 2 years, because it takes the time to produce the material, I will have the results and I'll be happy to share them.
Okay, thank you.
And just to add that, as you talked to when you start to think on this epigenetic effect, I remember well at the Congress that we had in Lisbon, the European Congress in Evolutionary Biology, where Carlos Herrera, which is like a master on ecology, that he had a seminar on epigenetics, and people were kind of surprised of seeing Carlos Herrera thinking about that on epigenetics, and people were there because it was Carlos Herrera giving it.
Yeah, it was controversial.
Like quite controversial seeing that such a renowned ecologist in Spain was thinking about it, and now it's everywhere.
Yeah, no, and I mean, it's such a shame that I didn't manage to publish this at the time when I actually got the results, because it would have been in the pioneering papers. Although, to be honest, like the possibility of actual methylome regulation, we only got it in 2014 when there was this paper about inbreeding depression in methylated and demethylated plants from Verheer or Verhoeven. They're both Dutch and I tend to mix them up. It's horrible, I know. But there was this amazing paper, like a very simple experiment, inbred, outbred offspring, and when you demethylate them, the differences simply disappear. That was so beautiful to see. So this is exactly what I want to do in Lamium, but with open and closed flowers and inbred and outbred offspring. Just takes a little bit of time.
So actually the changes that you are seeing in the populations are the result of the second part of the inflorescence stalk, right? So the base, it's predefined cleistogamous.
Actually, the period and the amount of constitutive cleistogamy can also vary.
Okay.
So it can only increase it, not necessarily decrease it, but I have data showing that constitutive cleistogamy varies between populations and between environments.
People at home. Yeah.
Yes, I see something in the chat.
That's my comment.
Okay.
And when I was introducing, I forgot about people on the phones. You said that the choice was
So how do we know that the flowers are predetermined 2 weeks in advance? in the 1970s, early 1980s, there was this amazing American plant physiologist, E.M. Lord, I think, that did a series of experiments on Lamium and amplexicaule, including exposing them to different environmental conditions at different times of their development and seeing when the production of open and closed flowers will change. So she did that, and I only looked at her results. She also modified the amounts of gibberellic acid the plants were receiving in a standardized environment, and also showed that if it's applied at the good time, in the good amount, it will also influence the production of open and closed flowers. I don't see any questions in the chat so far.
At home, anyone that would like to pose a question, please just go ahead and mute yourself and just
Or you can type them in the
or
chat.
write it.
I'm
Yeah,
fine
yeah,
with typing
yeah,
them
please.
in the chat
Yes.
as well.
So, really nice.
Thank you.
I was also very, it was very nice also to see that the timing of the sowing was huge impact, because we suspect that, well, yes, yeah, as well, with the experiments that we have. That's
Yeah.
interesting.
I kind of have a, let's say, latent experimental trauma from this, because now whenever people say, oh, but it's okay, you'll grow the plants in a greenhouse, you can just sow them anytime and then you can have up to 3 generations a year. I'm like, no, I want the right timing.
No, but the way is not here. We have several experiments with the occurrence in the greenhouse, and we are having some of them contrasting results. And again, I found out that we were sowing them in different times. And she explored that because we also had a reciprocal transplant in the field, where the timing of implementation is crucial.
Absolutely.
So maybe the contrasting effect, the result that we are having in the greenhouse, also depends on the moment of season determination.
Absolutely. Absolutely. Yeah, no, the timing is definitely crucial. And plus, like the species is also highly dormant, but with conditional dormants that can be broken down at different times. Like there are periods when you cannot break it down, but others where, depending on the condition, it can be broken down. It just complicates
It
it.
can be much more complex.
Yeah, no, it can always be more complex, but how about making it simpler? Because I chose this species originally thinking, you know, if the estimates of outcrossing rates work as planned, which they did, then it's very simple to study it for the rest of my PhD, and I was very wrong.
More frequent than we think.
Yes, absolutely.
There is no other question.
I don't see any raised hands or anything.
I'd like
Well,
to-
actually, yeah, sorry.
Please, no, go
Yeah,
ahead.
well, I would like then to, if there is no other questions, to thank you for showing up in presence or online. And if you have any additional questions, just send me a message.
Thank you very much.
I will, if you want to do the closing.
No, it's just thank you everybody. And if you want to revise the seminar, you can always do it through the webpage of Sociedade Broteriana and see it again at any moment. Thank you. Have a nice weekend.
Thank you.
That's it.
Bye.